Hello everyone who may be joining us from various parts around the world. My name is Justin Preece and I'm a faculty research assistant at Oregon State University in Pankaj Jaiswal's lab in the Botany and Plant Pathology department here. I work on the Gramene project in close collaboration with our colleagues at Cold Spring Harbor Labs on Long Island. And as part of that work in the Gramene project, I spend a lot of time focusing on the Plant Reactome project. The Plant Reactome is a data resource and a set of analysis tools that contains curated metabolic, regulatory, and signaling pathways in rice and orthology-based pathway projections for 66 other plant species with sequenced genomes and transcriptomes. And then we also provide tools for omics data analysis and interspecies pathway comparison. If you go to plantreactome.gramene.org, you will see this homepage, and you may be familiar with this from visiting the site previously. If you want to learn more about current functionality, you can go to that link at the bottom of the screen, tinyurl.com/Gramene, and it will take you to our YouTube channel where we post all of our prior Gramene webinars. This will include a webinar, more than one webinar that discusses current Plant Reactome functionality. But today, I'm going to talk about some new features that we have coming shortly in our upcoming release. Excuse me. And I'm going to spend a lot of time going through those new features to give you a sense of what's coming up soon. We're rather excited about this. It's a major software implementation. We have a lot of new things to show. So I'm going to cut over to a live demo and start walking through some of these items on our test server. Of course, it is a test server, so we're still in the process of polishing that up for release. There may be a few items here and there or labels that are need to be corrected before we put it into our live environment. If that's the case, just please bear with us. So going over to our test server, the home page is very familiar. It looks just the same as it did previously. There's a link through to the Pathway browser, analysis tools directly, a tutorial video that goes over functionality for our current live site, a link to the Reactome Wiki user guide, data downloads, and a contact us for an email address to get in touch. And then on the right-hand side, we have an embedded Twitter feed that just keeps you abreast of any recent developments on the entire Gramene project. Across the top, we have a menu that allows you to navigate to various areas of the site as well as a search bar. None of this changes in the new release, but what does change is the significant difference in the Pathway browser itself. If I click through that, you'll see our newest Pathway browser interface loading up, just to give you a little bit of a walk around this environment. And feel free, by the way, to interrupt with questions at any time on the chat interface. I'll keep an eye on that and watch out for questions as I go along. So to orient you to the Fireworks Pathway browser space, across the top we have a species selector. As I mentioned, our curated pathways are available in rice, but you can also use this drop-down to select any of our other 66 species across the plant kingdom. It also includes some lower order species as well. We'll stick with rice for the moment while we're giving this tour. There are some other icons available in the upper right-hand corner. You can go to analysis tools via this icon, which we'll come back to in a little bit. There's also a very nice tour that actually comes via our collaborators at the Human Reactome project. I should go ahead and mention now that much of the software was originally implemented at OICR, the Ontario Institute for Cancer Research, and at EBI, the European Bioinformatics Institute. They have done the bulk of the work on developing the software, and then they've been kind to work with us and share their software. We have forked projects in GitHub, and we've made our own modifications and then provided our own curated pathway data as well as our projected data to make the Plant Reactome. So you will see references in this test environment site to the Human Reactome project, and that's why you see that. So they have a nice tour available. If you want to click over to that, you can watch it. The information that they provide on their prepackaged video is actually 90–95% germane to the Plant Reactome. It can give you a good orientation to how to use the new site. There are options in the layout where you can remove the left nav or remove the details pane at the bottom, remove both at the same time to just focus on the fireworks display as well. Speaking of the left nav, this is what we refer to as the event hierarchy. Currently, we have our pathways organized into 5 larger categories: cellular processes, circadian rhythm, metabolism regulation, response to stress, and inflorescence development. You'll notice that the bulk of our curation work has been in the metabolism and regulation area, as is indicated by all of these nodal points on the graph that point to different pathways. You can expand the event hierarchy to look at pathways contained under these top-level containers, and then as you get further down into them, you'll also see the reactions listed. If I expand, for example, the metabolism and regulation, you can get a sense of how we have currently organized our pathways into broad functional categories: carbohydrate, cofactor, hormone biosynthesis, secondary metabolites, etc. If I open hormone biosynthesis, now we get down to specific pathways that you'd be familiar with from metabolic publication: jasmonic acid biosynthesis, acid signaling, etc. And then if I were to open any of these individual pathways, I would then see a listing for the reactions. All of these are linked and tightly coordinated with the actual pathway browser space, and I will take you into that in just a moment. In the lower hand portion, you've got a details pane, and most of this information for description, molecules, structures, expression, analysis, and downloads apply to the pathway view, so I will come back to that in just a little bit. The main view in the middle here is called the fireworks display, and fireworks is a description for, I suppose, how it looks as if these were exploding in the night sky. But it's a way of organizing our pathways so that you can see how that they are thematically grouped. If I, for example, zoom in on some of these, you will see more and more detail begin to appear. The largest circle in the middle represents all of metabolism and regulation, and then you get down into subcategories: secondary metabolite biosynthesis, and I can go even further than that and get down to an individual pathway like polyisoprenoid biosynthesis. And if I zoom in further, I can begin to actually read those labels. And these connecting lines do not represent any sort of biologically significant distance. They're merely hierarchical connections back to the categories that they belong to. As we move forward with the new fireworks interface, we're excited to reconsider how we have organized our pathway hierarchies, because we would like to be able to show functional connections across different pathway categories. So in the future, you will begin to see greater connections from pathways in one area over to pathways in another area that will allow you to navigate between them and see how the various cellular functions connect up. Let's go ahead and move into specific pathways so we can do some browsing. I'll select for now. We'll go over to ABA biosynthesis, and we'll stay in rice for the moment. ABA biosynthesis and mediated signaling. When I select this individual pathway, you'll see over in the fireworks diagram, it centers in on it. Then up in the upper left-hand corner of the fireworks browser, there's an icon here that allows me to open that pathway diagram. Once I select this, it will actually zoom in and load the diagram that represents our ABA biosynthesis and mediated signaling pathway, or collection of pathways, I should say, in this particular case. Now, when we're this zoomed out, you will see that it just gives broad boxes that delineate the areas of the pathway with no detail for abscisic acid biosynthesis and abscisic acid mediating signaling and their overlap and connection points. As I zoom in, you will see that detail begins to appear and those larger themed boxes disappear. And you start to see the actual pathway illustration itself, including metabolites that are involved, as well as the various gene products or collections of gene products, either in complexes or defined sets, and their connections to the reactions themselves. If I select an individual reaction, it will highlight everything that's involved in that immediate reaction. You'll notice as I zoom out, less consequential molecules, such as the metabolites, start to fade away, and it allows you to focus in more directly on gene product activity. I'd also like to point out that the Reactome platform allows us to localize our pathway activity within the different compartments of the cell. So in this particular example, you see that there is a plastic compartment delineating this area of the pathway illustration, as well as the larger cytosol. And they have defined, I think, something like 27 compartments. We have defined 27 compartments that allow us to represent, here's nucleoplasm, different areas of the cell and more accurately reflect the pathway illustrations. If I zoom back in and select a particular gene product or collection of gene products, let's just select this guy, PP2C here. I can begin to learn more detailed information about the pathway I'm looking at by opening up the details pane at the bottom. When I selected that element, you'll notice that it showed me all of its connective tissue in different parts of the excuse me, pathway diagram. There's a molecular listing under the Molecules tab. There are 35 proteins represented in this entire pathway, but while I've selected PP2C, it shows me which 5 of those are highlighted. For our curated rice pathways, in order to most fully integrate with the Reactome software platform, we identify them primarily by their UniProt identifier, but you'll see that the IRGSP identifier is also visible here. And then if you were to select a link, it will kick over to the UniProt interface where you can look at gene information about any particular protein of interest. For our projected species, we tend to link off to a variety of sources. We use 2 different sets of projection data, an in-house clustered, imparanoid clustered set that includes gene product data from multiple sources, but then we also connect to Ensemble for over 30 of our other projected genomes and use the compara orthology. I bring these 2 sources up just to say that in this linked area in the details pane, you will see other examples where it links out to Ensemble as co-hosted by the Gramene site or other external providers such as TER for Arabidopsis. So for the moment, you see this molecular listing. We see a highlight of those that are associated with just the element on the pathway diagram that we've selected, PP2C. You may be wondering about what all these various icons mean in the pathway diagram. There is a legend in the upper right-hand corner that will explain to you the difference between its representations of small molecules, proteins, complexes, sets, etc. So in this particular case, I have selected a set, and this set represents these 5 different gene products. It also illustrates for you the various types of connections and reactions that occur in a diagram. There are other tabs for other features. I'll go into these more as I move through the webinar, but this gives you basic orientation to the pathway browser space. The next thing I'd like to move to is this expression tab. We work with the Atlas group at EVI. They have curated a large set of multi-species baseline expression data across the plant and animal kingdoms. And we integrate with the plant baseline expression data in this site so that when you load up particular pathways, you can take a look at their set of canonical data and get a sense of what that baseline expression would look like. Let me give you a couple of examples of that. If I were to go over to a different pathway, I will use short day inflorescence, one of our more recently curated pathways. Obviously something that would not show up on the human reactome, but we have a need for. Let me close some things so you can more easily see that. So I go into inflorescence development. See, I can navigate by the event hierarchy without having to go back to the fireworks browser. And I select short day inflorescence. Now we've loaded a different pathway. And if I zoom in a little bit on that, if I were to select a particular gene on here, say EHD2, and it gives parenthetically its UniProt identifier for rice, then I could also go to this expression tab, and it would integrate and load for me the baseline expression data coming from Atlas. In this particular case, the baseline data for this experiment was organized by tissue type, and that tissue type is then correlated to this diagram of rice so that I can see where root, seed, shoot are actually occurring in this particular plant. Not all of our species have diagrams, but many do, and I'll show you an example of that with barley later on. You can also look at tissue samples at the seed level of the diagram as well. And these link off over to the Atlas baseline expression data set. So if you want to learn more about the Atlas data that's available as it pertains to your gene or genes of interest, you can link off to get that information. It's also downloadable in this space. Did my screen just stop sharing? Okay, just a moment. I'm going to reshare my screen. Okay, and we're back. Sorry about that. Lost screen sharing for a minute. Another example of the Atlas baseline expression data. Let me go ahead and switch species. I'm going to go back to our ABA biosynthesis model. You'll notice that it highlighted, since this is a subpathway, it will actually zoom in on the portion of the larger composite pathway. So if I start with ABA biosynthesis and mediated signaling, I can then drill down to just the biosynthesis portion. If I go there and then I go to the species selector, I can select another species such as Arabidopsis. We'll zoom back in. When we project, of course, we don't always have evidence for projection by orthology from our curated species to our projected species. So there will be sometimes gaps in the diagrams. But if just taking this example in front of me, if I select this complex off to the left-hand side of this defined set, you'll notice that there's no illustration on the left-hand side this time, and it's because this was a different type of experiment. I believe these are organized by ecotype. And so you have the genes associated with this complex and the baseline expression data that's available for that. And one more example, let's go to our circadian rhythm, another recent diagram. And I will skip down to ZMAs. Here's an example of baseline expression data organized by, as soon as I get zoomed in and selected on a gene, or a complex in this case, organized by time series, 10 days after, 12 days after, 14 days after. So it just gives you a sense of what Atlas provides to you via the Plant Reactome and then also over at the main Atlas website. If we can make a connection between the gene products that are listed in the Plant Reactome and the Atlas set of data, then we love to display it here. Let's see. Jumping around, I'm also showing off some of our more recent curation efforts. Response to cold temperature is a good one. And if I look at barley, for that, you'll get another example of as soon as I select a gene, illustration. So you get the illustration of the barley plant and more tissue examples in this area here. Okay, moving away from expression data, I'm going to talk about another new feature in the upcoming release for Plant Reactome, and that is working with interaction data. let me jump back to the circadian rhythm diagram, and we'll take a look at that in Arabidopsis. You'll notice that I shouldn't have closed that. Let me reload it real quickly. I'm reloading the circadian rhythm diagram to show you that it's attempting to find interactor data. You'll see that orange bar in the middle of the screen, no interactors found in intact static. We have a local database that comes with housed on the Plant Reactome server for interaction data, and we've got some upcoming experiments, not part of this release, but we're going to be working with some interaction data that we've collected in-house that we are going to provide via local database. But you can also connect to remote services. On the right-hand side, you see this blue bar. The middle icon takes you over to interactors. And inside there, you can select from a variety of local and remote sources to load interaction data overlays onto your pathway browsers. We just started working with this. I can give you an example of a preloaded example for Arabidopsis on the circadian rhythm diagram for a particular gene product, CDF1. Behind this gene locus identifier here. I'm going to move it off of the expression at the bottom just so we don't confuse the issue. And if I were to, in this middle portion, you see where it says Psychic right here. There's another service provided by Henninger-Jacobs Group at EBI. The Psychic service is a standard API interface that allows people to register their interaction data and then provide that to various software applications, including Reactome and Plant Reactome. You can select from a variety of providers to go get this remote data. This is not stored on our server. This is over at EBI. And I can look at their remote intact database of interaction data. And by selecting that, you'll see that it begins to load up interactors that are available for genes on the circadian rhythm pathway in Arabidopsis. Now, my experience is that this takes a few seconds to load. I have preloaded some results of the interaction data for exactly this example. So while that's loading, I can show you from a previous query what happens when it does find interaction results is it drops these little red circles next to the gene products for which it has interaction data. I can click on that red circle, and it will then pull up a hub and spoke diagram of the various connectors, things that interact with that particular gene according to this remote interaction data source. If I click on one of those, it will take me over to the UniProt site. So in the case of CDF1, it would jump over here and I could get more information about that interactor. You can also upload local data as well under custom resources at the bottom. In addition to going to remote sources, you could add your own, and you can do this via a file of just columnar data. The formats are explained in this pane at the bottom. You can get a sense of how that information is organized. I won't go into detail on that, but you can read about it yourself once we've released the site. You can also copy-paste your own two-column interaction data right here. You can also do multi-column. You can show strength of interaction with a numeric column as well. Or you can point to a URL where you have a downloadable file that has interaction data. So you can bring your own data to the site and use that. If you're really getting ambitious, you want to build your own psychic service, then you can connect to that as well. Something else, moving off of interactions now, I'd like to highlight is the improved visualization features associated with the analysis tools. If I close the interaction and I go up here to the top nav and I select Analyze Your Data, or Analysis pane, I have a couple of different tools that are available to me. Now these, the tools in general are not new. These are provided in the current release, but we do have some new visualization features that make them prettier and I think slightly easier to use with some of the color scheme options that you have available to customize your results. I'm going to start with a species comparison example as one of the two main tools. If I want, I can compare our reference species to any of the projected species in the Plant Reactome. I'll select maize for the moment. And when it does a species comparison, what it's doing is it's taking a look at the orthology data that we use to make the projections in the first place, and it then lists these in an overlay mode on the site. Let me go change the color scheme for some of these results, make it a little easier to see. Go. As soon as I select the one that I'm hoping to see. Oh, right. I'm still on Arabidopsis, so even though I've compared rice to maize, I have to actually go to the rice view in order to see those species comparison results. There we go. That's what I was looking to see. So when you do a species comparison, it will show you where it detects presence and absence of gene products by orthology from our curated species rice over to the projected species, in this case maize. The yellow represents where it has an orthology detection that it can make, and then if it's not yellow, for blue, for example, it does not have, this exists in rice, but it does not exist in maize where we're projecting to. If you move to a higher level of organization where we're just looking at the broad categories, it will also show you just by percentage representation in those categories exactly where it finds projected data by comparison. On the left nav as well, you can also see that it gives just a simple statistical count as well as a calculated FDR for the representation of that comparison. If I were to go into another specific example one more time, I can show you also some of the effects of changing the color scheme. So if you need to, for some reason, change the look and feel, say for a screen capture or for visualization for publication or something like that, you can change the color schemes that it's using to represent this information. Okay, the other main tool that comes in the analysis tools, again, not a new functional feature, but a new look and feel to it, is the expression overlay or data analysis visualization. There's a wide variety of kind of data that you can provide to this and then view as an overlay. I'll give a couple of examples today. Obviously, we're not going to be hanging on to the cancer gene census options on the overlay for the Plant Reactome. So we'll be removing that shortly. But I'll start with an example of using some preloaded metabolomics data. This is just a sample data set, and you'll see the molecular IDs on the left here from KEBI that represent the various small molecules that might be involved in metabolic activity. If I then click continue, I can specify whether or not I want to just see this overlay on rice or see it on all species, and whether or not I want to include the interactors that we have loaded in our local database. I'll stick to just rice for now. I click analyze, it does that work, and then it shows me where I can expect to find these metabolites. I will kick over to, let's just select something. Any of these are good options. I've got lots of metabolic activity. If I select flavonoid, for example, you can see that it gives me, because the data I provided was columnar, it could be time series, for example. It now gives me a navigation bar that allows me to walk through the different time points, or in other examples, it could be other types of columnar data. And I can see the affected change on the diagram where it has present the metabolites. As I mouse over these, you'll also see on the right-hand side, there's a little red bar in the gradient that gives you a sense of the level of metabolic activity that was recorded for these various items. I can also change the color schemes for this as well. So if you need a greater degree of differentiation, you can switch to Strosa bar or you can go back to the standard. Copper Plus is nice because it's a little bit brighter. I'm a bit partial to that one. With expression data, you can also work with, with the analysis overlays, you can also work with expression data, and you can upload your own data via file. I'll use an example of diurnal expression in rice. Just upload my own tab delimited file. There just has to be a header row explaining the columnar values. In this case, it's a time series. And then rice genes, MSU, and the numeric values. And by associating that file with the analysis, and ask it to continue, I'll project it to rice because in this particular case, I know that I'm dealing with rice expression data. It'll run that analysis, and then it will show you that listed by time series as well. I can go to another diagram, perhaps circadian. It will keep that analysis intact with the calculated data at the bottom, as well as the numeric values at each of the time points in the time series. And then here's that familiar bar where I can actually navigate through the changes in value. You can see some slight changes in coloration on the view. We might see more impactful changes if I switch over to the Strosa bar view. Yeah, there we go. So as I move through this data, you see some changes on the screen for the various types of expression data. You can zoom in, of course, and you can get details on any of these items. In the case of a complex or defined set, if there are multiple genes listed, it will bifurcate the item, or it will separate it into groups so you can see the different values listed inside there. You can also get details. There's this little blue arrow on the right-hand side that will give you the numeric values inside this viewable pane. And I can pin that pane to the screen. So as I move around to other things, that will still stay in place, and I can use that just to reference those values as I move around and look at other things. So that's the expression overlay. A couple of things that might be interested to of interest to any of the developers on the call. We also are now providing some new versions of APIs that you can code against in your own software projects to pull Plant Reactome data down. We have two main kinds. One is the content service, and the other is the analysis service. I'll just go over to the content service now. We have implemented a Swagger interface that allows you to see a summarized listing of all of the API methods that are available to you. Just a couple of quick examples. I can go down to the species category of methods. The content service will basically provide you access to all of the data within Plant Reactome. Not expression data, for example, that would be something that you would bring, but any of your curated or projected pathway data would be available here. I can do things, simple things, such as make a remote call to list all the species. Again, this is focused on software developers now, but you'll see here, even if you're not a software developer, I'm getting returned back a list of species in JSON format that I could consume in another application. Species list is pretty generic, though. You would probably want something else. Some other examples, you can go to pathways, and you can get a hierarchical list of the top-level pathways for a particular species, say rice in this particular case, and it will just list for you those top-level organizational pathways as they are right now: circadian rhythm, inflorescence, metabolism and regulation, etc. You can also query by ID. This is the one that would be of interest to more people, I'm sure. So if I go to the query category of methods, there it is, I can look up the kind of data that's used on the display just by putting in the ID value. Now, in some cases, these are actual gene names. In other cases, these are our Plant Reactome stable identifiers, which are available on the site by list and by bulk download. So if you wanted to, you could cross-reference these stable identifiers against their gene names and synonyms and then use these values to pull back data. I'm just going to grab a particular piece of data I know is associated with maize, and it looks like in this particular case I got a gene. I won't break into the data too much, but it's related to phosphorylated CCA1 and the expression thereof. So you see how you can pull down the very data that's used on the site via API and consume it yourself. That's the content service. The analysis service is the other main API in the new release, and it allows you to get, say for example, you've uploaded your own user data. There's a token associated with that data, and you can grab that token off of the URL at the top and then pull it back into here and actually download Plant Reactome's calculations of your data against the Plant Reactome database, and then use that again in your own application. That's a pretty esoteric use for most people, but it is available. Something else you can do here, however, is you can look at our pre-indexed analyzed data. You could pull a value by identifier. Here's an example of where I could do that with an actual gene name. And it will just retrieve values for me that represent that give me a sense of the overrepresentation and calculated statistics, p-value, FDR, etc. For this gene, and it happens to appear in circadian rhythm in this particular case. Last two things I would like to show you have to do with the searchability and access of our data. If you just go to the site for non-programmer access, for general use, there is a search bar at the very top, and you can search for values. Say, I just want to see in general, what does Plant Reactome have on ethylene? It will provide suggestions, but you can just use the keyword that you've typed as well. And it will give us results. It's faceted results on the left-hand side. You can narrow this down by species, if you like, or by type of result, proteins versus reactions versus pathways. So if I narrow this down to pathway, or say I'm interested in maize, not rice, I can go select Zea maize, unselect rice. So now I'm seeing ethylene keyword search results, pathways only for Zea maize, and I see those results over here. If I were to select one of those, I get more detailed information. It gives me that stable identifier. These, by the way, they're called stable for a reason. Release to release, this number will not change. We will maintain a version history of changes to the underlying data, but you will be able to reference these stable identifiers release to release. And then it gives you a location in the pathway browser where the search information, where these results appeared, and I could click through to there and it would actually navigate in maize to that pathway search results. Another quick example, if I wanted to say I'm looking for a gene of interest, say TOC1, I can go and look at those results, say just in rice, take that top protein hit. It tells me TOC1 appears, where it appears, oh, circadian rhythm, big surprise, right? And then it also tells me at the bottom which species it has been projected to. So if I wanted to look at this result in another species, say Arabidopsis sternensis, then it will take me to those results, and then I could link to the circadian rhythm pathway or reactions in that pathway over on the pathway diagram, and it would take me over there, again, within this particular species. These search results are also integrated over with Gramene, and let's see if I have those preloaded, yes. So you can actually do a similar search over at search.gramene.org, and they have already loaded Plant Reactome data over there, and say if I want to search for something like jasmonic in their various results at Gramene, because of course they're covering much more than just plant pathways, covering genomes and grow processes and many other things. I do see results under Plant Reactome for various things, and I can go look at those results, and Gramene will have taken its genome data and tell me across its phylogenetic organization of its various species where there are genes associated with jasmonic acid biosynthesis in those species, organized by within the various chromosomally delineated genome listings. And at the bottom, you'll see those gene details, and you can get into more information on the Gramene side. So it's just an example of how we're well connected with them. Going forward, we've started to play with, and this is a future functionality that's not going to be available quite yet. We are developing, Andrew Olson at Cold Spring Harbor has been working on even tighter integration using some new features where we can embed widgets that represent pathways on the Plant Reactome just by calling them. It can pull them in. And also the fireworks diagrams. We can embed those in search results. So if I were to look for orthologs KAO2, for example, and I want to find them in Plant Reactome, it will show me where those orthologs are across the various species in Gramene. And then there will be in the future a Pathways tab. This data right here is coming from the Plant Reactome via those APIs I showed you earlier. And if I wanted to select any of those individual results, we can now use those widgets to pull down those diagrams directly into the Gramene search results. SAM1 is another good example in Arabidopsis, where it shows how SAM1 appears in multiple pathways. And I can select any one of those particular examples, and it will load that location of that gene activity within the Plant Reactome. Gives you an idea of the power of these integrated graphical APIs, and we look forward to providing more and more features at Gramene from Plant Reactome as we move forward. We're also going to be doing more work in the future on bringing in our own interaction data onto the local Plant Reactome database, so you'd be able to use that and reference that without having to upload your own interaction data. And we've also recently been working on pathway gene associations based on phylogeny, and we've been performing analyses on the genes present in curated pathways for a majority of our projected species in order to examine gene family evolution and speciation. So more to look from on that work in the future. I would want to make sure to mention that we are an NSF-funded project, so thank you, National Science Foundation, and I want to extend a very hearty thanks to all of our great coworkers at Cold Spring Harbor Laboratory and our collaborators at NYU, the Ontario Institute for Cancer Research, and EBI. I particularly like to highlight some developers who made a lot of the software that you saw today work so well. Antonio Fabregat-Mundo and his team at EBI is responsible for a lot of the visualization and pathway browsing tools that you saw. And Joel Weiser and Ram Hua and Wan Ming Hu for their excellent work on the initial data structure and the work on the Reactome Curator tool, as well as maintaining the servers that we use. We're also working closely with various collaborators at individual genome projects like tree-based, maize, GDB, bar, soy-based, peanut-based, and we look to continue to deepen those relationships going forward. We use a lot of their genome data to build our projected orthologies on the Plant Reactome, and we're going to be working with them more in the future